Lehigh University. I'm the second act today. And I'm going to kind of talk about something much different, related, but much different than Ivy did. And as you can see, my title of my talk is Observed Limits to Darwinian Evolution. And you might ask yourselves, well, what's a talk like that doing in a conference here, sponsored by Ratio Christi, a Christian group, and this is actually going to be a purely scientific talk. And the answer, I think, is obvious that, of course, questions about evolution and where we come from and so on, how did everything get here, how did life get here, overlap significantly with religious, theological, philosophical concerns. So my point here is just to give you kind of a basic understanding of some new data in science that relate to this question and then can be used to help evaluate claims about more important things. So before I start, I always give a little warning that I'm not talking for my colleagues here. I'm not officially representing my university, in case you thought I was. My mother disagrees with me. This is just me talking here. So the primary question that I'm going to address is this one. What is our best evidence of what Darwinian processes can actually do? Ever since Darwin published his book on the origin of species back in the 19th century, great claims have been made for Darwinian processes about how it can explain life. And also, a lot of people have been very skeptical about it. There's been a lot of ink and a lot of arguments on the topic back and forth. Darwinists claiming, it can do this, and skeptics saying, no, it can't, and then retorting, yes, it can, and no, it can't. So, but in science, one would really like to have, data that bear on the question. And so what I'm going to argue is that the best evidence we have to assess the abilities of Darwinian processes arguably comes from studies of malaria, both in the genetic changes of humans and in the parasite, the malarial parasite itself. And these data have only become available within the past 20 years or so. And the reasons that it's malaria gives us a good insight into evolution is for two reasons. There are detailed genetic studies of both humans and the malarial parasite. And the reason, of course, is that because malaria sickens us. It's a disease that affects a lot of people, and we're very interested in health issues. So it's been studied in very great detail. And the second reason that it's very pertinent is that there have been a large number of organisms that have been in this evolutionary race between malaria's desire to eat us and our desire to not be eaten by malaria. There have been perhaps 100 billion people who have had malaria over the course of history, since in the 10,000 years that the modern form of malaria has been around. There has been an astronomical number of malaria parasites themselves, because in one person who is sick with malaria, you can have 1 trillion or so malarial cells. And just to refresh your memory, this is kind of the how malaria gets transmitted. The malarial parasite itself, which is called Plasmodium falciparum, is actually a single-celled creature. And it lives in the gut of mosquitoes. And the poor mosquitoes, they've gotten a bad rap, but they're victims just like we are. They're not happy with being infected by the malaria either. Nonetheless, they get infected, and then they bite a human, and some of the malarial cells can come out with the mosquito's saliva, and they travel to the liver of the infected person. and they kind of hang out there for a couple weeks and develop a little bit. And then after a couple of weeks, they come into the bloodstream of the person. And one malarial cell, right here, this little gray dot here, will invade a red blood cell, and it will enter into it, and it will eat the hemoglobin inside the red blood cell. And with the energy and materials that it gets from eating the hemoglobin, it makes more copies of itself. It makes 20 copies of itself. And then, after it's eaten everything it can, it bursts out of the red blood cell. And now there are 20 malarial cells, each one of which attaches to another red blood cell. And in the next cycle, there are 400 malarial cells. And in the next cycle, 8,000, and so on. It goes up exponentially. And they can eat a large fraction of a person's blood. And that's oftentimes what causes a person's death. And although we've forgotten about it in this country, we used to have malarial infections as well. But it's still, it was eradicated in the United States back after World War II, but it's still a major problem for world health. It still kills 1 million people a year, primarily young children. And here's a photo of a modern mother and child. The child is sick with malaria. And this has been repeated maybe billions and billions of times, maybe up to 1 trillion times in the history of humanity. And if you think about it, if a poor child gets infected by malaria, if they had had a mutation occurred in their genome, which gave them the ability to resist the malarial parasite even a little bit, then their chances of surviving a malarial infection would be better than perhaps children in their neighboring village. And they might survive, and if they do, they might grow up, have families, and their children might inherit that mutation. And it turns out that that process has happened a large number of times in the history of life on Earth. And the most famous mutation that helps a person resist malaria is called the sickle cell mutation. And here's a map that that shows where malaria occurs in light blue, where the sickle cell gene occurs in pink, and then their overlap in purple. And you can see that for the most part, the occurrence of the sickle cell gene is limited to regions where there is malaria. And that's a good evidence that it gives them some resistance to malaria. We won't go into how that happens. But we will ask the question, well, this is great. This is shown as in most biology texts as a great example of Darwinian evolution. Mutation in the face of some selective pressure, the mutants can survive better, and then they increase in the population. And it is a very good example of Darwinian evolution. But the question is, what is that mutation? What exactly is happening at the molecular level to allow this to occur? And that is where the progress of science in the past a number of decades has allowed us to see what exactly is going on during evolutionary processes. Before, our ability to track down mutations at the molecular level, at the DNA level, we could only see that some organisms survive better than others, and evolutionists would then run with that and claim that that could explain pretty much all of life. But when we look closer, we see a different story. It turns out that the sickle mutation is a change in the hemoglobin of a person. Now remember, the red blood cells contain hemoglobin. It is the hemoglobin that the malarial parasites eat. So it turns out that a change in the hemoglobin can help humans resist malaria. But hemoglobin is a protein that consists of two types of chains, one called the alpha chain and another called the beta chain. And as you may remember from your biology, proteins are made up of units of amino acids hooked together to give a large structure. It's kind of like snap lock beads, if you've ever used those when you were a kid. You put them together in different orders, and that's what proteins are made of. The sickle mutation is a change in just one out of 300 or so amino acids in the hemoglobin molecule. It's a change from glutamic acid here in the number 6 position of the beta chain to a valine. And we won't go into how that helps resist malaria. But the point I want to make is that it is a small, tiny change in a pre-existing system, a pre-existing protein. Now it turns out that mutations in DNA, changes in DNA, come in different flavors. Some mutations are called substitution mutations, where one kind of DNA subunit, called a nucleotide, is switched for another. And that can lead to the switch of an amino acid in a protein like hemoglobin for another, as in a sickle mutation. But there are other kinds of mutations as well, called deletion mutations, where some DNA that was there is thrown away. Or insertion mutations, where new DNA is added to existing DNA. And other types of mutations as well. So it turns out that in history, not only the substitution mutations occur, but all sorts of other mutations as well. And some of those other mutations also allow a person to resist malaria. And let's just go through some of them that have been discovered in the past 20 years or so. Some of them are also mutations in hemoglobin, just like the sickle mutation is. And we should also point out that that one change of the glutamic acid for a valine helps people who have just one copy of the sickle mutation that they might get from their mother or their father, if they have a normal copy as well, that is one that doesn't have the mutation. But if they get 2 copies of the sickle mutation, then they have sickle cell disease. And I'm sure everybody here has heard of that, and that itself is a lethal illness, as lethal as malaria itself is. So that mutation, that switch of that one amino acid, is kind of a desperate measure. It's not leading to some improvement on some absolute scale. It's not leading to anything sophisticated or elegant, like the molecular machinery, other molecular machinery found in the cell. Another thing to notice is that this mutation does not occur in the immune system, which is ordinarily the system which is responsible for defending us against microscopic invaders. How about some of the other mutations? Well, there's another mutation called an alpha thalassemia mutation. And in that, the gene for the alpha chain of hemoglobin that we saw on the last slide is broken and thrown away. And for some reason, that gives a degree of immunity to malaria to the person who has that. Nonetheless, it can lead to anemia, and it results in a broken gene. Another mutation that is selected for in the presence of malaria leads to something called beta thalassemia, in which the gene for the beta chain of hemoglobin is broken, deleted, thrown away. Again, it leads to anemia and a broken gene. Another mutation that can help leads to something called hereditary persistence of fetal hemoglobin. Before birth, babies make a different kind of hemoglobin called fetal hemoglobin. And usually that's turned off right around birth and adult hemoglobin is made. But for some people in malarious regions of the world that have sickle hemoglobin, the control regions of the DNA that turn off the production of the fetal hemoglobin at birth are broken. And that turns out to help a little bit in resistance to malaria. Nonetheless, they all have in common the fact that they're breaking functioning pieces of DNA that were already there. In a different gene, one for something called glucose 6-phosphate dehydrogenase, which is a protein called an enzyme, which helps kind of do the housekeeping work of the red blood cell. There are a lot of different mutations and deletions in the gene for this protein, which break it, which cause it not to work anymore. And for some reason, again, that gives resistance to malaria. The gene for something called band-3 protein, deleted, that helps in fighting malaria. But kind of like with sickle cell, if you have 2 copies of the deleted gene, if you get a broken gene from your mother and your father, then a child dies in utero. It's never born. Duffy antigen. If you break the controls that turn that on in the red blood cell. And all of these are helpful in resisting malaria. So we can, just from this data, we can conclude 3 things. And the first is that much Darwinian evolution proceeds by breaking old genes. Not only that, but breaking a gene is a lot easier to do than to make a specific change in a gene which you might imagine would lead to an improvement. And the reason is that, for example, in hemoglobin, there are hundreds of different places in the gene that you can delete something or mess something up. There might be only 1 or 2 places where a change would lead to an improvement. So there's a lot more. It's the old way, or the old idea that there's a lot more ways to break something, to break some functioning machine, than there is to improve it. Take your, say, cell phone or something. Lots of ways to break it. Not so many ways to improve it. Another point is that very, very few of possible mutations are helpful. Now, I said that in the course of history, in the past 10,000 years, there have probably been hundreds of billions of people exposed to malaria. And in those hundreds of billions of people, there were not only occurring the mutations that I listed on the screen, lots and lots of mutations would occur in different genes just by chance. Nonetheless, apparently none of them could help. None of them helped at all. The ones that we see when we go out in the field and sequence the DNA of people who are resistant to malaria are just those handful, maybe 5 or 6 types of mutations listed on the last slide. So of all possible mutations in all possible genes in the human genome, only a handful can help resist this deadly disease. So very, very severe selective pressure, nonetheless, only a handful of possible helpful mutations. The last point is that those random changes that we see occurring for malaria are incoherent. And by that, I mean they're not adding up to anything. They're not putting together some bigger system. For example, deleting the gene for band-3 protein does not help improve the sickle cell mutation, the glutamic to valine transition in hemoglobin. they're utterly unrelated. So there are 100 people in a clinic. The malaria has never seen a atovaquone before. You give all of those patients the drug, and 30 of them, the malaria spontaneously develops resistance. This is much, much more, about 100 million times faster than resistance arose to chloroquine. So the question is, why? Why can malaria develop resistance to the newer drugs a lot faster than it did to chloroquine? Well, it was unknown for a while, but studies did show that with the newer drugs, resistance could arise when only one particular amino acid in one of the proteins of malaria would change. And it turns out that there is about 100 million or so of those amino acids, but there is about 1 in 10 to the 12th, a trillion cells in each person. So that means there's a fair chance that in each person infected with malaria, one of those malarial cells is going to have that unique mutation that allows it to develop resistance to the newer drug. How about with chloroquine? Well, it was only last year, where is the date on here? 2014 or so. This is published in 2014. That the molecular basis for the resistance of malaria to chloroquine was discovered. There was a lot of complications, but the long and the short of it is that two particular mutations in one particular protein of malaria had to both be present for resistance to occur. If only one was present without the other one, then it was ineffective. Not only ineffective, it actually hurt the malarial cell, because this was a protein that it needed. And one of the mutations by itself would actually cripple the function of the protein. But when the other one appeared with that first one, then it could confer resistance. But what are the odds of getting those two mutations at the same time? Well, we have a lot of experience with that. If you played a lottery, suppose you had to, there was a lottery where you had to pick a number from 1 to 100. What's your odds of picking the winning number? Well, your odds of picking the winning number are 1 in 100. Okay, but suppose you had to pick two numbers between 1 and 100. What are the odds then? Are they still 1 in 100? Are they 1 in 101? Are they 1 in 200? No, they're multiplied because they're independent choices. You put your hand in, you pick one ball out, and then you put it in, pick another one out. So it's 100 times 100, or 1 in 10,000. In other words, when you have to get 2, the odds against being correct go up exponentially. Now, if there are, say, 100 million or 1 billion different mutations possible in the malarial parasite's genome, the odds of getting one of them are 1 in, say, 1 billion. And that's what we saw for the atovaquone, for the resistance that was easily developed. But if you have to get 2, then that's 1 in 10 to the 9th, 1 in 1 billion times 1 in 1 billion. Or that's roughly 1 in 10 to the 20th, with a few other factors considered. That's an astronomical number of chances that have to occur. Literally astronomical. There's something like 10 to the 15th or so stars in our galaxy. So the odds of getting 2 of them require an enormous number of chances. And that's why chloroquine was so effective for so long. But what does that portend for Darwinian processes? It means that whenever you need more than one mutation to occur to have some improvement, say to make a molecular machine or to make a protein or something. You need an enormous number of organisms. You need a long, long time. And if you need more than 2, if you need, say, 3 mutations to occur, then it's effectively impossible for random processes to get you there. And let me just tell you that that getting just 2 amino acid changes is pretty, is not much. I mean, that won't get you very far in biology. So we're coming up against a very, very stringent limit to Darwinian processes. And I want to emphasize this point, that what we're looking at is data. So what I'm telling you here is this is not an argument anymore about what Darwinian processes can or can't do. These are observations about what it does and doesn't do. And when we look at it and see what it doesn't do much. And let me re-emphasize that the selective pressure on these malarial organisms was intense. It was being eradicated by chloroquine. Any mutation that would help would be selected. And all we get after an astronomical number of chances, 10 to the 20th, are 2 little amino acid changes in 1 pre-existing protein. Another point to make is this. Some people say, well, yeah, we're smarter these days. We know that there are other processes in biology other than Darwinian ones, where you have random mutation and natural selection. But in nature, when the malarial parasite was trying to survive these drugs, nobody was around telling it what sorts of processes it could use to try to defeat the drugs. Nobody was saying, you can only use Darwinian processes. No, what we do is we challenged it with the drugs, and after a long time, we saw what it did do. In Darwinian processes, the random changes, the astronomically improbable random changes, helped a little bit because there were an astronomical number of parasites that could chance upon those 2 mutations. But other possible processes which evolutionary biologists have invoked, such as self-organization, self-engineering, symbiosis, nor any as yet undiscovered process helped the malarial parasite. So we do not have to guess about what would happen in this case. We see that nothing really helped the malaria except raw numbers of organisms that gave it enough chances to come together to change those 2 amino acids at one time. Okay, well, let's look at another system. Malaria, as I said, I think is our best evidence, but let's look at some other systems as well. One is a laboratory evolution experiment that's been being conducted by this man whose name is Richard Lensky. He's a professor of microbiology at Michigan State University. And 25 or so years ago, Lensky decided to grow some E. coli bacteria cells in a flask in his laboratory. And so he grew some one day, and the next day he came in and he took a portion of those cells and put it into new growth media and allowed them to grow another day. And each day they would go through 5 or 6 generations. And then he transferred them again and again and again and again. And it turns out that after 25 years, they've gone through about 50,000 generations. And there have been about 100 trillion E. coli cells in total throughout that time. So this is a wonderful example, or a wonderful opportunity to look and see what evolutionary processes can do when we're just looking. Now, it turns out that he's growing the E. coli in the lab, and you can ask yourself, well, yeah, but you know, their environment stays the same. You know, why would they evolve? Well, it turns out that evolution doesn't stop. If some mutation came along which gave one bacterium a little bit of a reproductive advantage over the other ones in the flask, it would leave more offspring, and in the next generation, its offspring would leave more offspring, and pretty soon it would take over the flask, and it would dominate the population. Then if another mutation came along that gave an advantage, it would take over. So it's not, you don't have to have a changing environment to see evolutionary processes. Even in a system like this, you can see it. And the long and the short of it is that we see things almost identical to what we see in humans under pressure from malaria. We see some mutations which are advantageous, but they're all breaking genes. Here's a paper from about 10 or so years ago where they looked at a line of cells which was improved in the sense that it was growing faster than the original line of cells grew. But nonetheless, he noticed that the expression of a particular gene had decreased after a number of generations. And it turns out it had decreased because the gene had been deleted. Apparently it didn't need this gene in his conditions. And so rather than wasting energy trying to make the gene and make the protein products for which it codes, it threw it away. It's kind of like being in a ship that's sinking on the high seas. And if you can just make it to port, you'll be okay. So what can you do to help that ship stay afloat? Well, you can take maybe some sophisticated machinery on the ship, maybe some advanced computers or other machinery, and throw them overboard. And that will help you float along. But that does not explain where the computers came from in the first place. Okay, so that's what we're seeing. So this ribose operon was deleted. Another mutation which gave a pretty big evolutionary growth advantage to the cells pretty early on was the deletion of the genes that made the bacterial flagellum, which is like a little outboard motor that bacteria use to swim. A very sophisticated molecular machine, it was thrown out, and that helped the bacterium in these circumstances. And through the years, he looked at other genes, which are listed here, and saw that mutations in a lot of different places in the genes were advantageous, helped the cells grow. But it turns out that all of these were destroying the gene or breaking the gene. As I said, there's a lot of ways to break sophisticated machinery, not so many ways to improve it. And so what we see repeated here with another organism is the same thing. Here, this is a bacterium. The malarial parasite is a eukaryotic cell, single cell. Humans are multicellular eukaryotes. Nonetheless, they all do pretty much the same thing. They make little tweaks in pre-existing systems, or they throw things away. And that's what we see Darwinian processes doing. I reviewed Lensky's work and a lot of other laboratory evolution experiments in a paper I published about 5 years or so ago now, in Quarterly Review of Biology. And I thought up this nice, succinct first rule of adaptive evolution. What to do first if you are an organism that has to adapt to new circumstances. And it is this: break or blunt any functional coded element whose loss would yield a net fitness gain. And that's just the same way of saying throw away anything that will help you float longer. break anything that's no longer needed. Okay, so those are the systematic studies. For the last part, I want to just look at a few stories that have been in the news media relatively recently, in the past couple of years, demonstrating the power of evolution as the stories go. Oh, I'm sorry. Here's a little lesson slide. What observation demonstrates about random mutations? Of those mutations that affect an organism, about 99% are detrimental. We knew that already. Most mutations hurt. Most mutations do damage. But no evolutionist was worried about that because the organism with the mutation would then die off. It's no longer, that mutation is no longer in the gene pool. What we are now discovering, what is new, is the second point, that of even beneficial mutations, the great majority break genes or degrade function. This is a huge problem now for Darwinian evolution because these mutations aren't going to be gotten rid of. They'll spread in the gene pool. They'll be stuck. They'll be fixed forever. Okay, so back to the recent stories about mutations that seem to be helpful. A story in the New York Times just about a year or so ago talked about a new mutations that were identified in a gene that protect against heart attacks. You know, our modern society, we have, you know, eating habits that are not necessarily healthy. So somebody who's born with a mutation that helps you resist, say, heart disease from our unhealthy eating environment will have an advantage. Here's one which gives an advantage mutation in a particular gene. But if you read further into the article, it turns out that the mutations destroy the function of a pre-existing gene. They're not building anything new. They broke something that was already there. Another story that appeared in the Times, here's the technical paper, talks about mutations that protect against type 2 diabetes, which we get more often in our culture because of what we consume. But again, they were loss of function mutations, which is a fancy way, the technical way of saying they broke the gene. Okay, so another set of helpful mutations that break the gene. Another cute story that was in the news was a mutation that is widespread in domestic horses, which allows them to trot more smoothly and which is preferred by riders. And so those horses are taken and bred for horses that have that trait. And the genetics behind that particular trait, which gives them this smooth riding gait, were investigated, and was seen that it's due to something called a nonsense mutation in a particular gene that makes a component of nerve cells. A nonsense mutation is another way of saying a broken gene. These days, we have a very great ability to sequence DNA and whole genomes of different organisms, not only the human genome that's been sequenced, but now anything that's of interest is being sequenced. And one thing that was sequenced a little while ago was the genome of the brown bear. And recently, some folks sequenced the genome of the polar bear. And it's thought that the polar bear is derived from the brown bear over several 100,000 years. But interestingly, when they looked to see the differences in the genomes between the brown bear and the polar bear, they saw that over half of the mutations that made the polar bear were due essentially, were predicted to be functionally damaging. In other words, conclusion that you can make whole new species, things that impress us as being somewhat different, by breaking the genes of a pre-existing species. Aren't they cute? Dogs are oftentimes cited by folks who want to talk about the power of evolution. They say from a wolf ancestor, you know, some thousands of years ago, we domesticated dogs, and look how they have varied. Look how many different kinds of breeds, different characteristics of dogs that have come about in only a short period of time. And it's true, and it was impressive, but only in recent years have scientists been able to look at exactly what were those mutations, what were those things that were being selected. And again, I'm not going to go through it all, but it turns out that the great majority of the genes, the mutations that give dogs these varied characteristics are due to the breaking of pre-existing genes. I'm going to skip over the same result for plants and the same result for bacteria and get to the point here. And what have we learned about Darwinian processes from this recent, the past 20, 25 years of advances in science? First, we see that random mutation is a bull in a china shop. Genomes are full of functioning instructions for functioning systems. And when you make a random change in that, the odds are great that what you're going to do is break something. And that's what happens. Nonetheless, sometimes breaking things help. And here's one image that I want to leave you with at the end of this talk on what observation shows Darwinian processes doing. And that is this. Now this is an image that I got off the internet. And what it is of here's some South American military men, soldiers. And they, here's a convoy. They had gotten word that there was a drug growing operation out in the forest. And so they were coming to raid the drug operation. But the drug lords got wind of the impending raid, and they blew up the only bridge leading to their compound. Now, from the point of view of the drug operators, this is a beneficial mutation. This is a helpful change. Now they're not going to be invaded by hostile organisms. This is exactly what happens in many cases in biology. One of those mutations which helps resist malaria literally is in a protein that ordinarily is in the membrane, the skin of the red blood cell. And the malarial parasite can bind specifically to that protein and use its molecular machinery to get inside the cell. And the mutation blows up that protein, blows up that bridge by which malaria gets inside the cell. Nonetheless, blowing up a bridge does not tell you how bridges are built. And the process that blows up bridges is not one that you would look to construct other kinds of systems too. Okay, so this has not gone over well with many folks in the biology community, and some people have taken issue with some of the points. So if you want to look at these points in more detail than we have time to go into today, I've posted some papers, some formal philosophical and scientific papers on my website at Lehigh. I've also maintained a blog for a while, at least at a site called Uncommon Descent. But most often, I've posted replies and comments at the website of the Discovery Institute. So anybody who wants to follow these things up, this is where to look. So thanks very much for your attention. I'll be glad to address questions if anybody has any. And let me just say that you also have cards in front of you. We don't have to do the cards to ask questions right now. I'll be glad to take some questions by hand. But if you have questions that you can't ask right now because of time, please write them down and hand the cards in to the Ratio Christi folks here. Or even if you just want to have a comment or something that you don't want to say in public for various reasons, please just write it down on those cards that you have in front of you and you can pass them to those folks. But in the meantime, if anybody has a question, I'll be glad to respond. Yes, sir?
Sure. The class I took many years ago, maybe 20, was basically postulating the same thing you were. But the question I had was, has any of the Darwinists come up with anything as a positive new gene that's not breaking gene? Do they have any examples of supposed positive evolution based on that theory?
Well, there's lots of examples of what they point to as new stuff that seems to occur in organisms after they branched off for something else. But the question is not, is there new stuff? The question is, how did it get there? And you can't tell just by looking and seeing that, well, there's a new gene there, which occurred, say, 1 million years ago in this line of organisms. The power of this is that, it's going on in real time. This is what we're looking at. So this is what we would expect from this sort of process. In the stuff such as the malarial and human evolutionary processes, in the experimental work of Richard Lensky and other folks, all we see is this kind of degradatory or sometimes minor, tweaks around the edges of pre-existing systems. We do not see anything looking remotely like it was going to make a new gene or build something that was not there already. Yes? Yes, sir.
Actually, you answered part of it, but I have another one. So, has anybody looked at the genes of the early humans and compared them to the genes of the current humans? Are more of the process, is it more degradation, or has there been some evolution?
Okay, that's a good question. Early humans, these days, you can even sequence the genomes of Neanderthals, from a bone you find. Sometimes there's residual DNA left over. And the long and the short is that it's hard to tell. It's hard to tell. You can see changes, but you see a lot of changes. You don't know which ones made a difference or not. You can see some changes in some genes that are already, we know what they do in modern day humans, but you don't know if they did the same thing back then. Yeah, that's the problem. It's hard to track down exactly what they're doing. Yes, sir?
Are Neanderthals humans then?
I'm sorry?
Are the Neanderthals human?
Well, that's a debated question. Are Neanderthals humans? And that's not my specialty, but some people say, yeah, they were just, a branch of Homo sapiens. They were just a branch of modern humans. Other people say, no, they were a separate branch of humanity, a separate species. And, it's hard to resolve those questions. Paleontologists joke with each other that they say there are two kinds of paleontologists. There are the lumpers and the splitters. And that's when they get fossils, somebody who wants to name a fossil after himself will say, this is a new species. Somebody else, his rival from another lab, will say, no, that's just a member of another species. There's lots of variation in species that we see today. This doesn't look like something new. So, in a large sense, it's a matter of opinion whether they're the same species or just a variation of within the human species. Yes?
Question. You talked a lot about mutations that break genes. So, do you have a thought on how would you define an unbroken gene?
An unbroken gene, well, an unbroken gene is one that has an identifiable function, and it's performing that function. For example, in hemoglobin, hemoglobin is a protein that kind of binds oxygen in our lungs, where we breathe in air, and carries it to the peripheral tissues and drops off the oxygen there. The genes for the two parts of hemoglobin, the alpha and beta, code for the proteins. When you have thalassemia, changes in that gene occur so that no protein is produced which is able to carry oxygen. So I would call that a broken gene. It's just like, it's the same thing as saying, I've got a bicycle. How do you define a broken bicycle? Well, if the wheel fell off and you can't use it to transport yourself anymore, then we'll call that broken. Same thing with the proteins, pretty much. Yes?
Okay. So, two questions. First of all, just to clarify, so currently today and maybe like the last couple of decades, there has never been a recorded example of a gene that was a mutation that was beneficial and not destructive, right? Was that what you said?
Well, I didn't say that exactly. There have been ones that, as I call it, kind of tweak a pre-existing gene. That is, a gene maybe makes an enzyme, makes a protein that catalyzes a chemical reaction, say, helps to break down glucose into other components. Sometimes if you change a particular amino acid, it can do that faster or slower. It still does it, but changes the speed a little bit. You can see mutations like that. But a large fraction of mutations that we do see in experiments like these do break the gene, and that it doesn't do what it did before anymore. Not all, but there haven't been any positive mutations that are doing new and different things and leading to complex systems. If you look at the cell, I haven't been able to talk about it here, the cell is full of very sophisticated molecular machinery. And there's nothing that seems to be on, even the first step of its way towards anything like that.
What's the ratio of tweaks to just destructive?
Well, I would say,
in terms of magnitude.
in terms of what?
Magnitude. Like
Magnitude.
1 to 10.
1 to 10, yeah, maybe even 1 to 4 or something. 1 tweak to 4 broken genes, something like that.
Okay, and I guess my last question is, sorry.
No, that's fine, that's fine.
This is not really that related, but is genetic engineering changing, destroying, or creating? Like current genetic engineering?
Current genetic engineering.
Just out of curiosity.
Well, it depends. In some ways, it can put new things in organisms, but almost always it uses materials that it gets from other organisms. Now, a fun example is there's a gene for something called green fluorescent protein. And there's these sea creatures, this jellyfish or something, that have the ability to make a protein which fluoresces, that is, gives off green light. And scientists have been able to isolate that gene using this DNA technology that has been developed. And they can take it and, say, put it in a mouse. And now you can put the mouse in the dark and shine some light on it and it glows green. And that's cool. And, you can buy some in pet shops at various places. But has it developed something new? And arguably, no, you just took a pre-existing thing here and you put it in another organism. But, I won't say that we intelligent designers, we humans, wouldn't be able to make something new that wasn't there in nature already. But I would say it won't be by Darwinian processes. It won't be by unintelligent processes. Yes, sir?
Does research be relevant to cancer research in the sense that why does the human body not differentiate between whether cancer is a good mutation or bad mutation, because it seems to happen anyway? So could we use some sort of medicine, like the chlora or something, to block certain genes themselves
Sure.
to prevent that?
Absolutely, yeah, all of this stuff. You know, the first thing you write in your grant proposal whenever you ask for money to do any experiment is how it's relevant to cancer research. But yeah, it's true that the more we understand of how biology works, the better we'll be able to understand when it goes awry. And new work understanding how cancer cells kind of lose control of their ability to grow properly is leading to the development of new drugs, new effective drugs, and so on. But the mutations that occur in cancer are pretty much mutations that you see elsewhere too. They break things that are already there. They break controls that normally keep it in place. Or they break controls of the body that allow it to defend itself, and so on. So this would be is helpful, but the point is that it does not show the power, does not show any constructive power for Darwinian processes. Yes.
Just another question, not really cancer, but you see a lot of mutations are based on breaking. Is there another process that helps regeneration? Because like the entire length of the strand doesn't stay constant, it's going to get shorter through time.
It gets shorter.
Is that part of life expectancy at all? Or
Well, we're talking about two different processes now. If you're talking about an individual organism, you know, usually the DNA stays the same in an individual organism through its lifetime. If you're talking about a species over many generations, then they tend to lose DNA. You do get mutations that can add DNA in the sense that you can get a piece of DNA which duplicates itself, say. So now you've got two copies where you had only one. And those, we see things like that happen, especially in turns out in cancer cells where such mutations can occur, and in other places too. But the point to keep in mind is that if, you know, suppose you had a book and, you know, you photocopied a page 10 times, and you stuck those 10 pages into the book, how much more information, you know, how much has the story changed since you added those 10 pages? It doesn't change it at all. What you need is, you know, explain where the new information, the new story came from. And for that, you generally need an author, not just a photocopying machine. Yes, sir.
Well, I don't want to take it away from the genetics stuff. This is quite unrelated. I already asked her for her opinion. But the question is, from the biological point of view, how come Jesus is a dude? Where did he get a Y chromosome since he was born by a virgin?
Oh, okay, okay. Yeah, I thought that was the question you were asking. It was that you that asked that question?
Yeah.
Okay. So I think the question about Jesus being a dude meant, why is Jesus a male if he was born as a virgin birth? You know, from where did the Y chromosome come from that Mary, you know, that somehow Jesus obtained, although Mary did not have a Y chromosome? And the short answer is, I don't know. I don't know. Because the flask in which the bacteria were growing was shaken. So they didn't have to swim to get food. Okay, it was essentially stirred up for them. But yeah, if you try to take those bacteria and ask yourself, you know, how likely is it that they'll regenerate a flagellum you know, I'll be glad to bet you, you know, a fair amount of money on the question. And that has not been seen in any reported experiment. Yes?
Mike, after the selective pressure of chloroquine is removed, would those bacteria that causes malaria lose their ability to be resistant to it? Would it go away? Would that broken gene come back in generation?
Well, it turns out that gene is not broken. The gene that they have, it's tweaked. I would call it tweaked. It has 2 amino acid changes. But interestingly, that experiment has been done because in some regions of the world, chloroquine became so ineffective that nobody used it anymore, and they used different drugs. And it turns out that it did not come back. The strains that had the changes in their in that protein did not revert, but other unchanged malaria that were at the periphery of the area that were using chloroquine would now invade the area where it was no longer used. And they were actually fitter, they were better at reproducing than the ones that had the thing. In the absence of chloroquine, the original malaria were better at surviving than the mutant malaria were. In the presence of chloroquine, of course, the mutant's better, but in the absence, the original is better. And they come back and take things over again. So there have been proposals that, well, if you get chloroquine resistance malaria for a while, you just, you know, use another drug for, you know, 10, 20 years, and then, you know, cycle back to chloroquine, and then cycle back to the other drug, and then cycle back, and maybe that can manage things a bit. But yeah, even with those 2 changes, it was not an absolute improvement in the ability of the malaria to survive. It was only relative to the fact that they were being poisoned. They had to get rid of this poison somehow. So it was not it was not anything that they would have done in the absence of this drug. Okay, now I get to ask you questions. Just kidding.
That's a question.
Oh, I'm sorry. Yes.
So we were talking about how you could have a mutation by, you know, a little tweak where you switch an amino acid. You could have a deletion, and then you also mentioned that there's a chance that an additional amino acid could insert. Could you talk a little bit more about that?
Okay, there's, yeah. Well, DNA, as you know, is what's called the instruction manual. It carries the information for making the stuff that actually does the chores of life, the chores in the cell. It makes proteins, and it's the proteins which actually go off and carry oxygen and, you know, catalyze chemical reactions and so on. All of the mutations have to occur in the DNA, and when the mutation occurs in the DNA, it can then affect the protein that it codes for. So DNA gets copied each generation. You probably remember from your biology. The 2 strands of DNA come apart, and machinery copies a replica of each strand, and then the 2 double strands go into the separate cells. When this thing is copying, though, sometimes mistakes happen. It means, you know, it should put in one nucleotide, one particular subunit of DNA, but accidentally it puts in a different one. That would be a substitution. Sometimes it's supposed to put in a nucleotide, but it kind of skips over and doesn't put in the nucleotide and kind of joins it to the one succeeding that. And then you have a deletion. There's no longer this nucleotide. Sometimes it puts in 2, where it's supposed to have put in 1. That's the insertion. Now, it turns out that the information in the DNA is read 3 units, 3 nucleotides at a time. 3 nucleotides specify 1 amino acid. So the 3 in a row might specify a glutamic acid, the next 3 specify a valine or so on. And the machinery reads these 3, and then skips to the next 3, and then skips to the next 3, and so on. Now, if you have an insertion, that's what's called a frame shift. So that it reads these 3, but now the next 3 are different from what it was. The 3 after that are different from what they were. It gets out of order. So that generally destroys the gene. It produces a protein in the sense it's a string of amino acids, but it doesn't have the right sequence to do anything useful. And so it's a broken protein. Same thing if you leave one of them out. That's a deletion, but it will change the frame of the reading machinery, and you'll get a non-functional sequence. Sometimes, though, if you put in 3 nucleotides in the DNA, or take out 3 at a time, then you produce a protein that's just 1 shorter, or 1 amino acid longer, but it's pretty much got the same sequence as the starting one. That happens on occasion, too. So those things can happen. But the power of this data is that all of those things could have happened in, say, Richard Lensky's flasks. You know, we weren't telling the bacteria not to make certain kinds of mistakes. Or they could have happened in the wild with malaria. We were telling the malaria what to try and what not to try. And presumably, all of those things did happen in some statistical, amount. But none of them helped, at least in these examples. It's not to say they couldn't help in some particular circumstance, but they don't seem to help much. And there's a lot of circumstances, apparently, too, that they can't help in. And they seem not to have any particular, beneficial effect that something else doesn't have. So with this data, you can rule out, or you can come to a judgment on all sorts of evolutionary processes, not only the ones that have been postulated, but even ones we haven't thought of, if they're, if they're due to random processes in nature plus selection. Suppose they're, oftentimes people object to intelligent design folks like myself by saying, maybe there's something going on in nature that we can't even imagine. And I say, well, that you can't imagine. But then, I don't want to alienate them. So then I say, well, yes, but, that doesn't matter. If there were, all we're looking for is results here. And the results are, very unimpressive, to say the least. So we rule out not only Darwinian processes, which most scientists, would defend, but a lot of scientists also will say, no, there's other processes going on. We rule out, or we get data that are relevant to any process that depends on chance processes, undirected processes, plus selection. Okay, get new people here. Yes, sir.
Yeah, I'm wondering if the work with the E. coli could be used to construct a time frame of what you might expect to see given the most generous old Earth views. I got there by, I think it was 20,000 generations. And so I put that in human terms, given 20 years or so, maybe 400,000 years.
This is actually 50,000 generations,
Okay.
so over 1 million years. Yeah.
Right. So you think that work could be used to construct a time frame of what one might actually expect to see over big
Yes.
country?
Yes, you certainly can. And as a matter of fact, I did that 10 years ago in a book called The Edge of Evolution. That's one of the 2 books on these topics that I wrote. You can, not only the E. coli, but in particular the malarial work on the malarial parasite itself. The argument is that it took 10 to the 20th organism, 10 to the 20th malarial parasites, to generate 2 crummy mutations that were helpful. And if you do calculations based on standard assumptions about how long the Earth has been around and about how many cells have been on the Earth, there have been about 10 to the 40th cells on the Earth. Now, if you know exponential, arithmetic, 10 to the 20th times 10 to the 20th is 10 to the 40th. So if you needed, say, 4 mutations, 2 and 2, independent mutations to get some selective effect, you're essentially, you would require all of the resources on all of the Earth for all of time just to get that one effect. So it puts, these types of experiments can put severe limits on what to expect from Darwinian processes. And to talk about the E. coli stuff in particular, it's interesting, yes, you mentioned what's the number of generations in terms of large animals like humans, whose generation times is 20, 25 years. And what Lensky has done is gone through at least 1 million+ years worth of evolution. And random processes are supposed to have taken, a chimp-like organism and turned it into a human-like organism in 6 million years. And we get an equivalent of 1 million years, and all we see are broken genes in the E. coli example. That should make somebody very skeptical that such a process could produce something much, much different than what it started with in the case of primates.
How valid would the argument that was in a test tube?
That's right. People have made that argument that, well, this is an artificial system. It's in a laboratory. Lensky gave it foodstuffs, keep it at the right temperature, and all sorts of stuff. Well, you can respond to that in 2 ways. First of all, that is, one is that, well, evolution happened nonetheless. He got lots of mutations. And if a constructive mutation had come along instead of a destructive one to allow some cell to reproduce faster than its neighbors, it would have taken over the flask. So that doesn't matter that it's in a laboratory. The second thing you can say is that, well, we have that in E. coli in a laboratory, and that matches what we see in, say, humans in history when they had to, when they acquired mutations in their DNA to resist malaria. We see the same sorts of destructive mutations, the same tweaks. Same thing that we see in malaria, the plasmodium itself. We see just a few crummy little changes in much more organisms than we see. So it's not just this one. We see it all over life, from bacteria to simple animals to complex animals. That's what we see, and when you see it that way, you should infer that that's probably what it does. Yes, sir.
What's the average lifespan of a bacteria?
I'm sorry?
What's the average lifespan of a bacteria?
Okay, well, lifespan is not quite the right word. It's, the generation time, because bacteria just kind of split. Usually they don't just die. And with the E. coli, it's a couple hours in the laboratory. For some bacteria, that just kind of veg out in nature, they can kind of reproduce just once a year. But in the best circumstances, when they're given all of the foods that they can eat and optimal conditions, they can reproduce in 20 minutes. Lensky's stuff is, a few hours. So it's a wonderful system for studying, how much changes can occur per generation instead of, using mice, reproduce every couple months or something. So we can get a lot more data with this system than with larger organisms. Yes, sir?
You've been talking about mutations that are basically replication type mutations, where the radiation exposure is pretty much constant. But how about over geologic history when we don't know really how much the cosmic ray fields change at all?
Okay.
Total mystery?
Well, yeah. So the question is, what if mutations happen more quickly because cosmic rays increased? Or you don't even have to worry about that. You can say, what if the replication apparatus of a cell wasn't as good as it usually is and made more mistakes than it usually does? And actually, that's what happens to the bacteria that Lensky worked with. After a few tens of thousands of generations, he kept separate lines, a dozen different lines of cells, so essentially 12 replicate populations. One by one, they all became what are called mutator strains, which means they lost the ability to correct mistakes in their DNA, and their mutation rates increased by about 100-fold. And they don't do any better than the other ones. And he hasn't investigated them or reported on them much in his papers, but you would expect them to actually go into what's called error catastrophe, where bad mutations accumulate so fast that you don't have enough time to get rid of them. Suppose an organism, a bacterial cell, was born, each cell was born with 3 deleterious, bad mutations. Well, when it replicates, if each cell was born with 3, then there's no way to get rid of those. And in the next generation, it'll get 3 more bad mutations, and 3 more, and then 3 more. And it's been likened to, say, photocopying a page on a bad photocopier, and it produces smudges when you photocopy it. And then you take the photocopy and you photocopy it, and you get more smudges. And after a while, you've lost all the information that was there to start with. So increasing the mutation rate is not really a viable way to get around this problem. Okay, I think we're good here.